right, our next speaker is Dr. Whitlock from the Boston Children's Hospital, and she'll be presenting brain protection by transamniotic stem cell therapy, TRASCET, in a model of intrauterine growth restriction, IUGR.
Good morning, and thank you for the opportunity to present our work. We have nothing to disclose. Intrauterine growth restriction, or IUGR, is a common disease present in up to 8% of pregnancies and the second leading cause of infant mortality following prematurity. It is caused by placental insufficiency, which is due to uterine natural killer cell activation to a pro-inflammatory and cytolytic state, leading to downstream inflammatory signaling and placental ischemia, which results in poor nutrient transfer to the fetus. IUGR is divided into two forms: symmetric, in which the whole somatic growth of the fetus is equally affected, and asymmetric, or brain sparing, in which the brain is significantly larger than the rest of the body and perhaps in normal percentiles, while the remainder of the body is in the lowest percentiles for growth. Among the many long-term sequelae of IUGR, the most concerning ones affect the brain. Children with a history of IUGR have higher rates of cognitive delay and behavioral health disorders than their non-growth restricted peers. This neurologic sequela is thought to be due to increases in neuroinflammation, increased neuronal apoptosis, and decreased myelination, which affects the fetal brain development in utero. Transamniotic stem cell therapy, or TRASCET, is a minimally invasive means of treating congenital disease developed by our lab. It involves the procurement and ex vivo expansion of fetal-derived stem cells and followed by their injection back into the amniotic cavity in large numbers. This can augment their role in inflammation and fetal wound healing, especially when MSCs or mesenchymal stem cells are utilized. A central component to the TRASCET principle is the fact that these donor MSCs traffic robustly through the placenta, and from there they reach the fetal circulation and even the fetal bone marrow. This placental trafficking has significantly expanded the applicability of TRASCET, with a recent study showing that it can improve fetal weight in a model of IUGR, likely by reducing placental inflammation. In light of that background, we hypothesized that TRASCET with MSCs, which are known to possess immunomodulatory and anti-apoptotic effects, might also provide fetal neuroprotection in the setting of IUGR. To test this hypothesis, we used a well-established model of intrauterine growth restriction, exposing rat dams to cyclic hypoxia in the final week of pregnancy starting on gestational day 15, and then dividing them into 4 groups. One group had no further manipulation, while the others underwent intraamniotic injection of either saline or banked amniotic fluid-derived mesenchymal stem cells. In order to further augment the anti-inflammatory properties of the MSCs, a subset of the injected cells were primed by exposure to select pro-inflammatory cytokines prior to their injection. All dams were sacrificed at term around E21, when their fetuses and placentas were weighed and the fetal brains were procured for multiple analyses. Prior to injection, syngeneic MSCs were obtained from Lewis rat amniotic fluid and confirmed by flow cytometry. The MSCs were primed by exposing them to interferon gamma and interleukin-1 beta for 24 hours, pushing them towards an anti-inflammatory phenotype before their amniotic injection. At term, multiple morphometric analyses were measured, and multiple ELISA analyses were performed on the fetal brain samples and for multiple markers. Inflammation was assessed with TNF-alpha and IL-1 beta, apoptosis with BCL2, and myelination with myelin basic protein. Here you can see our gross morphometrics, which confirmed that the fetal weight was significantly reduced in the untreated and sham groups and normalized in both TRASCET groups. Placental efficiency, which is defined as the ratio of placental to fetal weight, was also reduced in the hypoxic groups, but significantly higher than the sham in both TRASCET groups. TRASCET was associated with an increase in fetal brain mass compared to the untreated and sham. The brain to body mass ratio was significantly increased in both TRASCET groups compared to the untreated and sham groups as well. Interestingly, the ratio was actually higher than normal controls, likely reproducing the brain-sparing or asymmetric form of IUGR in these groups. Levels of pro-inflammatory cytokines TNF-alpha and IL-1 beta were significantly decreased in the TRASCET groups compared to the highly elevated levels seen in the untreated and sham. TNF-alpha normalized in the TRASCET groups, but IL-1 beta normalized only in the prime TRASCET group. Anti-apoptotic BCL2 levels were significantly lower in the untreated sham and TRACET groups, but normalized in the TRACET prime group. Myelin basic protein, or MBP, was significantly lower in the untreated and sham groups, but normalized in both TRACET groups. In conclusion, in addition to reversing core effects of intrauterine growth restriction, transamniotic stem cell therapy with mesenchymal stem cells, particularly if primed to an enhanced anti-inflammatory state, provides fetal neuroprotection in the setting of experimental intrauterine growth restriction. This is likely due to modulation of neuroinflammation, reducing apoptosis, and increasing myelination. Further study working towards clinical application of this novel strategy for IUGR is certainly warranted. Thank you to my co-authors, to our sources of support, and to you for your attention. I look forward to your questions.
Thank you. Questions from the group? I have one thought. I think this is really elegant work, and I'm excited to see the direction it goes. Do you feel like there are any ways you can kind of tweak your experimental method to even further enhance the benefits that you're seeing, or do you feel like at some point you just sort of reach a limitation for what this therapy might be able to offer?
Thank you. That's an excellent question, and it's something that we actually are really actively exploring. I think the priming of the cells is something that you saw Dr. Labus is also doing, and it's something kind of new to our lab, and I think that we could further optimize. Right now, we're priming them for 24 hours with cytokines previously used by other labs, but I think that we could explore further enhancing that anti-inflammatory effect, because that really does seem to be the source of improvement in this model. We have further mechanistic data that we're looking at the placenta itself, and we're seeing that the inflammation in the placenta is certainly modulated by these prime cells as well, and so it's something we can definitely further optimize.
Thanks. Other questions? All right, we'll look forward to further work. Thank you.
Thank you.